Please introduce yourself.
Hi everyone. Our group name is Alien Species, and it's so because we chose an invasive species to work on. My name is Parth. I'm a botany honors student at Bhaskaracharya College of Applied Sciences at the University of Delhi, and my teammates will also be introducing themselves.
Hello everyone, I am Neil Kumar. I am pursuing Bachelor's of Technology. I am pursuing Bachelor's of Technology in Software Engineering from Delhi Technological University.
Hello everyone, I am Dipanshu from Sri Venkateshwara College pursuing BSc Biological Sciences. Currently I am in second year.
Hello everyone, I'm Rahul, a third year student from Ramjas College pursuing BSc Life Science.
Okay, so here we basically would use the title demonstration of extraction and reuse of data in invasion ecology using the tools that we have learned in the past couple of days using the Plazi and the semantic climate toolkit. I will be explaining the introduction portion regarding the particular plant that we chose. As we all may be aware, invasive plants are commonly recognized to have very severe ecological impacts at the ecosystem level, as they can alter structure and function and trophic structure, also resource availability, and they are known to contribute to degrade biodiversity and natural landscape. And basically, non-native exotic alien species, whenever they invade a particular area, they can pose a serious threat to the native plant communities and the population dynamics of that particular area. The plant that we chose is Lantana camara. It is considered an alien species in India as well, and is also recognized as an alien species by International Union for Conservation of Nature as one of the world's 100 most invasive species. And it is also considered as among the world's 10 most worst weeds. Now, Dipanshu will be explaining the principles that
So the usage of fair data is important in the sense that we have a lot of data around lantana. So we need to, so it's invasive basically, and we must know the phytochemicals that are responsible so that we can study allelopathy if there is, or how it is affecting the micro population around. So having the open data and AI readable data or accessible data is very important so that further studies can be made. And we have tried to like start something which we can lead forward. So about objective.
So now moving into the core of our study objectives, our first objective is to find out the global distribution of the lantana camera across different countries in urban and peri-urban areas, and by using the available published scientific literature. And our second objective is to know the reason why. I mean, beyond moving the just location, we have to know about why it is so invasive. So what are those phytochemicals that is like might be associated with this for its high success. So now moving into the method.
So let me walk you through the methodology which we have used in this. So first the data was retrieved country-wise distribution, and we used three keywords. First is invasive species, second is urban, and third is lantana based on Plazi article collection statistics. So then we did automated literature review using the semantic climate toolkit, in which the latest 50 papers were retrieved based on those three keywords: invasive species, urban, and lantana, and then a word cloud was formed based on that country data and phytochemical compounds. And based on those word cloud, we have integrated these to make a network graph, which was shown over here. And so the main motive is to analyze and reuse these literature data to make some meaningful inferences.
Among the inferences, first we have the screenshot of the Plazi article collection statistics website. Here we selected certain criteria: number of articles, keywords, affiliation of those particular authors, along with the number of treatments and the collecting countries. Within the collecting countries, it could be from the first glance, it could be seen that lantana is widely distributed among the tropical countries, and it's clearly an invasive species there. However, all of this data could be made into a well-readable format and further analyze. So right now we just looked at it from a very preliminary perspective.
So now here we have used the semantic climate toolkit to identify to find out the countries. So what we have seen here is that the size of the country name represents the frequency of the name that is being mentioned in the scientific literatures. So what we have found here is India, Pakistan, Ethiopia, and a few other countries uh, have been mentioned uh, in the available scientific literature data.
So we made network graphs of the data that was available with the different keywords that we use. Something interesting that we noticed was we changed the keyword from lantana camera to just lantana to see what difference we can see. So the one we see on the left hand side had the keyword lantana camera. So countries were there, but we couldn't find multiple okay, so the blue one is the paper and the pink one is the countries and the green one you see here is the compound. Com- no, it's the compound. It's the compound. Yeah, it's everything is lantana because the lantana was keyword here. The keyword here was lantana camera, and we could find only two compounds, acetone and camphor. So that was not true. So we changed the keyword to lantana, just lantana, not lantana camera, and we got this hair ball type of. So we need to analyze this further. So this is yeah, the orange one, yes, and the it's yeah, sure. We need to analyze this more. Yeah, yeah, we did. I don't know. We need to check this further. So we need to analyze this. So this was something. So this is just of lantana. So we could see there are more compounds which we can see. So we need to trace back. So what other lantana species has what other compounds and what allelopathic they hold other than lantana camera. How many others are invasive? So this leads a way to that.
Next, below this, we have the fourth figure, which where we made the word cloud for different phytochemicals that were available for the keywords that we use, lantana camara, and of course the last 50 papers only. And we cleaned it up a little because there were also other chemical compounds like alcohol, for instance, acetone, which are basically solutions that we were using. So we could get all of these. It needs to be analyzed further about the role of all of these, like the linoleic acid or things like acetate or euglynol. Maybe they play a role in the ecological success of lantana's invasion.
Okay, thank you. Well, one thing just to pick up on what Dr. Gitanjali said regarding figure 3. Uh, it's good, but please remember, all figures need to be self-sufficient. Like that, along with their captions, of course. The captions is included when I'm saying that. Uh, even if you don't put in the image, you need to describe it in the caption what each color is, which is fine for now. But in the future, yeah, please remember to do that. Will help you a lot. But yeah, do you have any more specific questions? I'll come back to.
No, not specific questions. I was just go to the treatment bank results. Yeah, I think this is very good, and this is something that I'm telling about when I was talking about methodology in the other groups, and this was a very nice solution because since we are here learning how to query, as I said many times, it's good to know how to query. So here with the results, since you have the fields there, we can see your search. So this is very nice because you have at the same time the results and what did you choose? Yeah, to make this query. But just as Felipe said, you had to take care of saying everything, the figures, thinking about that there will be in Zenodo, so they will receive an own DOI for extracting data and all that. And another good thing, first time we saw bibliographic references, so this is nice also, nice, very nice. When we are talking about being fair, we must have all the bibliographic references, so good for this.
You know, just still in within the screenshot, this first figure, it is still important to say which parameters were used, even though we can gauge what they are from what you're seeing right now. It's because we are used to that. It's important for replicability so that other people can reproduce exactly which fields you chose. So normally you can describe it or have another screenshot of that, but normally you would describe, I chose collecting countries and show individual values. That you need to inform that, otherwise people are like, oh, what should I do? Okay.
Yeah. And another thing that's very good is if you could go through, like there are not so many fields, so you could click and then see in one of them, like 2, 3, if they are in GB, if in the wrong place in map. So this is good for you to check. And then you could just open this same query and then you could see the map and then you see the errors that are coming. This is kind of the dashboard feeling. But again, we are being picky here because we really like the results. So congratulations.
Spelling of objectives. Which one of you will tell me? Okay, नहीं बताना पड़ेगा. No French, just the objectives, spelling. Run along. Very good work. I'm very impressed. What is the next thing you could do with all this? You've just learned the tools, and this is just one day's work. But what if I gave you a month or call you for 2 months to do an internship in the lab? What else could you do? Or you could pick time, have have the emissions changed. Yes, that's one thing. What could you do for urban research? इसमें मुझे urban दिखा नहीं है कुछ भी.
So we, like as I said, we can take the geospatial distribution, how it has been.
No, could you filter down that I don't want all the papers that came when I gave Lantana and urban and invasive and XYZ. I only want the research done on Lantana or the evidence for Lantana emissions in urban cities.
So that's what we did now.
You already filtered?
Yeah, we put the keyword urban there. So
No, no, no. You put the keyword, but you get much more than the keyword. That I saw. But like you just saw the millets, the team on millets explaining that you give a keyword for millets, but much else comes. So when you give a keyword urban, you don't assume that only urban will come. You have to look at the table. Did you use the semantic tool? Then in the first set, you will see the table on the paper titles. Some paper titles, just humanly reading them will make you realize that this is not truly urban research. Then you have to do some more focus on that to only take those papers which really reflect urban work. Because Lantana, lots of work is going on in national parks in the countries, in different countries, isn't it? Yeah. So how would you make sure that this is urban research only and not in protected areas, right? So think about that also. What else could you think? You tell me what else you could do if you had time.
Ma'am, if I have time, I will analyze and recreate that network graph by editing it and modifying it so that we can gain better insights from it and it will help us in further studies.
Very good. What would you do extra?
Ma'am, if I get enough time, then I will definitely go deeper into it and then see like what's lacking inside it. And
everything's lacking inside it. This is one day's work. So what would you do extra? No vague answers. I don't want ChatGPT answers. Tell me real stuff.
I will do like whatever the published literature is available. Then
today, how many papers you looked at in semantic? 50?
50.
Is that the right number? Do you think 50 papers only have been published for urban Lantana? You could expand that.
Yeah.
But we have 4 scientists standing in front of a good budding scientist. Good work.
Thank you, ma'am.